And Carmen is here. I walked in and I looked at the slide, and I don't think those are the people I thought were speaking. You shouldn't be so modest. You know,
No, just
again, as long as you spell your name correctly.
that might be the problem, not to risk
Yes,
it.
I realized this morning that that wisecrack probably went completely over the head of most people here who are significantly younger than I am, which is most people here. In the good old days of glorious starstruck Hollywood, when the standard line from the stars in regard to the publicity and the gossip columns and so on is that I don't care what they say about me as long as I spell my name, they spell my name correctly. So that was the source of the wisecrack. But in any case, wisecracks aside, we're pleased to have Carmen Molina-Paris from the University of Leeds, and she will be telling us a story about two receptors and two ligands.
Yes. So good afternoon. Thank you for the chance to present the work. And I need to do justice, if nothing else, to the people really that have made this possible. So first of all, the other way around, I never get this right. The work has been led by our PhD student, Luis de la Higuera Romero, and we've been very fortunate to have someone coming to Leeds with a background on something that we didn't lack, which is a bit more a probabilistic approach into mathematical modeling of biology, and this has been also done in collaboration with Grant Life, who's also a supervisor of Luis. So what's the background to these two receptors and two ligands? And I mean, I don't know if my talk might be sort of the odd one out, but in order to try, maybe Omer has given some, but I wasn't here. So we've been trying to understand some of the immunological processes and trying to model them. So when we started in immunology, our focus has been more the T-cell receptor. And then for a number of reasons, the T-cell receptor signal in immunology is called signal 1 for T-cells, and there is a signal 2 and a signal 3. So signal 2 comes from these other receptors that I'll be talking here about. And why we got into doing this was for two reasons. One, because these receptors are expressed in cells that are called regulatory T cells, and we were looking at them. But then more recently there was some new data that we happened to read from a group 2 years ago in Birmingham, now moved to UCL, on this interplay between these two receptors and ligands. And interestingly enough, sort of the classical understanding or the textbook immunology about these two receptors might be put to the challenge, and our job, which is really work in progress as I speak, is trying to understand this new mechanism and to model it. So that's what I'll try to do, and I've got, what, 40 minutes? Yeah. Right, so I'll provide an immunological introduction because I think it's important to understand what is the question that we are trying to address and where we're coming from. So the big question in immunology is this balance between whether you mount an immune response to something that could be a pathogen, yet the way you recognize these potential pathogens is by short peptides. And the peptides that can come from a pathogen are not necessarily that different from the peptides that can come from our own household-derived proteins, also called self-antigens. So this balance between recognizing the foreign, yet not mounting an immune response, is really a big challenge. And one way the immune system deals with this is by T cells are the ones that mount this adaptive immune response, and to that end they have receptors on their membrane of the order of 30,000 receptors called T-cell receptors. And the way the immune system, as I say, can cope with this immense variety of pathogens is by having a large diversity of these receptors of the order of 10 to the 6, 10 to the 7 different molecular structures of these T cell receptors. In order to ensure that the evolution of the pathogens and the genes encoding for these molecular structures of the T cell receptor, the genes encoding the TCR, the T cell receptor, are actually rearranged in a random way so that to avoid this potential problem. So even though this leads to a large diversity, by the fact that the rearrangement is random, one can actually get T cell receptors that will mount an immune response to your own self-proteins, and we're all aware of autoimmune diseases. So the way the immune system has evolved to really solve this problem of, again, the immune tolerance and mounting responses happens in the thymus by a process called central tolerance, and I will not talk about it. And then there are other solutions. When in the immune system one refers to central has to do with the thymus, which is an organ above your heart, and then peripheral is all the lymph nodes that we've got in our body. So when I talk about peripheral, is cells, immune cells circulating in the blood and moving from the blood to the lymph nodes. So another solution is by these peripheral tolerance mechanisms, and these are the ones that are encoding by these two receptors and two ligands that are the basis of the story. So given that really getting rid of all the potential T cell receptors that can mount an immune response to your own self-proteins, there are other mechanisms that are playing in the periphery, and these are mediated by two molecules. One is called the CD28 and the other one is CTLA4, also called co-receptors. And the idea is that even though these may seem to be redundant, CD28 and CTLA4 molecules, why have two, they actually seem to work as a checkpoint. And even though the signal 2 that I've referred to before is delivered by CD28, the role of CTLA4 has been maybe not so well deciphered, but it seems now that it's clear that CTLA4 really is essential into providing answers to this mechanism, but not by the classical textbook role that had been given to CTLA4. So I'll explain that in a minute. So it's important to talk to immunologists because you realize how important it is to know the immunology before you really get yourself to doing the modeling. So even though these two molecules, CD28 and CTLA4, as I've said, seem to be maybe redundant, if one knocks out CD28, what happens to these mice is that they cannot mount an immune response. So in the classical immunology, you need the signal from the T cell receptor, you need this co-receptor signal from CD28. So if you don't get a signal 2, then you don't mount an immune response. CTLA4 was assumed to be, in this classical textbook immunology picture, to be putting a brake on this signal 2 from CD28. Yet, when you actually delete CTLA4 in mice, what happens is that they actually become really sick and autoimmune. So, a rather different angle. So the question is, how can you have these two really opposing outcomes, sort of lack of an immune response or really, really a lot of immune response against your own self-proteins by two molecules that can bind the same ligands? And as I've said, really the mechanisms at the heart of this are yet being elucidating. So what is it that we know? So as I've said, CD28 combine these two ligands called CD80 and CD86. I'll be giving a picture in a minute. The idea is that T cells express the T cell receptor and these two co-receptors, CD28 and CTLA4. T cells need to talk to antigen-presenting cells. By definition, antigen-presenting cells are cells that present antigen. Antigen is the bit that binds to the T cell receptor. But antigen-presenting cells also express the ligands for the co-receptors, and these ligands are called CD80 and CD86. So in this picture, you have two receptors or two co-receptors and two ligands. So CTLA4 is not expressed by all T cells. Naive T cells, or those that have not seen antigen before, don't express it. And it's only activated T cells and regulatory T cells that express them at greater numbers on the membrane. And in this interplay between two receptors and two molecules that combine to them, CTLA-4 has a higher affinity for both ligands than CD28 and has this sort of classical inhibitory function that seems to downregulate T cell activation. So how does it work? What is this mechanism? So back to the picture. This is your T cell, this is your antigen presenting cell. So if we move from left to right, you need to have the T cell receptor signal first, then CD28 binds to either CD80 or CD86 and gives the co-stimulation signal too, and CTLA-4, again binding to either CD80 or CD86, will inhibit these two signals. And here, just for reference, the affinities of the different ligands and receptors that I'll be looking at. As I've said, CTLA-4 in this picture, when compared to CD28, is a winner in terms of the binding and unbinding properties.
But the CTLA-4 first has to be expressed, right? Is that
Absolutely, yeah. And then, yes, it's important to remember in this picture that CTLA-4, when a cell is naive, CTLA-4 is not expressed, okay. And that will be of relevance later on. Thank you. So something else that needs to be added into the picture is that whereas CD28 has a more sort of stationary profile in the sense that it seems to be sitting on the membrane, CTLA-4 is a molecule that traffics. And traffics, I know we're not doing space here, but just a bit of space for the time being. So CTLA-4 can be expressed on the membrane, but it's, even in the absence of ligand, is internalized through different vesicles called endosomes, and some of it might be degraded, some of it recycled, and also there's always turnover. And in fact, what happens with T cells is that when they get a T cell receptor signal, you get new synthesis of CTLA-4. So the overall numbers change. But in the absence of ligand, so without any stimulation, most of the CTLA-4 molecules are actually located intracellularly. So what happens when you signal through the TCR is both you have enhanced trafficking back to the surface and de novo synthesis. And as I've said, this trafficking property doesn't seem to be present in CD28, so that's already one difference. I am going to put here, so in this quest to try to put forward ideas about the different mechanisms that CTLA-4 may or may not have, immunologists have been thinking rather a lot. And they've sort of differentiated or separated between two types of mechanisms. One, which they call cell intrinsic, and I'll explain what they mean by cell intrinsic. So when they say it's a cell intrinsic mechanism, it means that cells that express the CTLA-4 molecule will have the effect. So one classical mechanism that seemed to be sort of the favored one was that CTLA-4 provides this inhibitory signal. So you have the signal from TCR, from CD28, which are signal 1 and signal 2, and CTLA-4 puts the brake. Another one, which of course is just basic ligand competition. If one receptor has a larger affinity, one may assume that it might steal ligand from the other competitor. And I'm not going to talk about other mechanisms. But they've also put a number of different cell extrinsic mechanisms. And in this language, a cell extrinsic mechanism is one in which the cell that expresses the CTLA-4 may not suffer a change, but it will affect a different cell. And I'll give just one example because it's the one that we'll be focusing more. And this is sort of the new immunology that is coming into the picture from this group in UCL led by Lucy Walker and David Sampson. So if you have a T cell that is expressing CTLA-4 in the membrane at large numbers, that cell can bind to an antigen presenting cell, and when the CTLA-4 molecule binds to any of the ligands, CD80 or CD86, it actually internalizes not just the CTLA-4, but the CTLA-4 bound to the ligand and stealing this little blue bit. So that when sometime later this APC, this antigen-presenting cell, sees a new T cell, it will not have enough ligands to provide signal for CD28, signal 2. So the idea is that this is a cell extrinsic mechanism, spatially and temporally separated from the first process. Spatially because it's a different cell, and temporal because these things may happen with half an hour of a difference. So the idea is that by having cells that express a lot of CTLA-4 in the membrane, when they contact an antigen-presenting cell, they will change it by, of course, depleting it of ligands. So when this APC may talk to another cell that requires signal 1 and signal 2, it will not deliver signal 2. And this is sort of the basis of the story that we'll be dealing with today. So, I mean, this is all in words. So the idea is that, in a nutshell, CTLA-4 will remove the ligands from the antigen-presenting cell by this mechanism of trafficking the whole receptor and the ligand. And as I've said, immunologically, this is called a cell extrinsic competition model. And of course, this APC being depleted from the ligands will not provide enough co-stimulation through the CD28 ligand. Important is that if one thinks about what is more relevant in vivo, because of course sometimes what happens in immunology is that the evidence that you get from in vitro is very different from what you may get in vivo. It seems that this effect of stealing this ligand, this cell extrinsic function, seems to be more relevant than the actual ligand competition in vivo. And again, you still have ligand competition, yes indeed, because the affinities don't change, but at the end of the day, you have yet this other mechanism by which you are sort of making the life of T cells and activation harder, instead of simply inhibiting the signal, which is a rather different mechanism. And important, keep this in mind, regulatory T cells express a lot of CTLA-4, so this is actually something that is rather relevant in the context of new cancer therapies that are being developed through this CTLA-4 pathway, and I'll explain what I mean by that. And again, this is just the picture, and now we move on to what we did. So what we did is, as I've said, thanks to Martin, who's been teaching us a lot of new things, and I'm learning quite a bit, put a little bit of a picture. And I think it's important to, before just going and putting both receptors and both ligands, maybe try to dissect things, particularly because for us this was new immunology that we're learning as I speak. So let's suppose my cell is only expressing one particular type of receptor, and the antigen-presenting cell that I am not drawing, I'm just putting the ligand there, but remember this ligand is not freely floating, it's expressed on the membrane of a different cell. So basically you have one receptor, one ligand, binding and unbinding reactions. The receptor free can be internalized, the bound complex can also be internalized. We are going to assume that the endosome environment is rather acidic. So as soon as this bound complex goes in, dissociation takes place. The free receptors in the endosome, this is R sub E, can be recycled back to the membrane. And as you can see is that if I start with a certain number of ligands, this process will be slowly depleting, well, slowly or fast, will be depleting ligands from this antigen presenting cell. So this is the model. These are the, more explicitly, the different reactions. So we have 5 different variables, and we've got 5 also different rates. And one can make use of a continuous time Markov process approach, in which, as I've said, we can have free ligand, free receptors on the surface, bound complexes, internalize free receptors, and internalize ligands in the endosome. And for the time being, because we wanted to do things pencil and paper, we are going to assume that the total number of receptors and ligands is fixed, and you have these two constraints. Initial conditions are going to be before at time 0 we have no bound complexes, nothing internalized, but the receptors may be in the endosome or in the surface, and our total number of free ligands is the initial one. So now we have 3 variables, and just to remember the order: free receptors in the endosome, bound complexes in the membrane, and ligand internalized, and this is the state space over which things are defined. And just for simplicity, we're going to look at the case, for simplicity, because that's sort of the immunology in this case, the number of ligands is smaller than the numbers of receptors, but one can do the other case. And we will put all this together. So these are the infinitesimal transition rates for this stochastic toy model, and these are the changes for state space. And the idea is that one question that one can ask ourselves is, given that there's this depletion of ligand, can we try to identify what are the timescales that are characteristic of depletion of ligand, say, to a certain percentage, right, half of the initial number. So in order to carry out with our analytical approach, what one can do, and as I've said, this is work that has been led by Martin in our group, is to arrange the state of the space of states in a clever way. So the way one can do that, and remember we had 3 variables, the numbers of receptors in the endosome, the numbers of bound complexes in the membrane, and the numbers of ligands internalized, is to say, well, first of all, if important to me is how much depletion of ligand I've got, I'm going to divide my state space by these sets or levels Lk, in which K determines how many ligands have already been internalized. So this index will tell me how much depletion has taken place. And now each level I can divide by sub-levels, in which I introduce this order R, or this index R, which tells me how many bound complexes I've got in my surface. So this is the sub-levels within Lk, Lkr, and these are the states in that subspace. I can do my counting, and I can see how many states I've got in each sub-level and how many states I've got in each level. And important is not just to classify them in a way that I can count them and order the states, but also realize that my transitions, and that's what we're looking at, my transitions between levels or sub-levels only occur between adjacent states. So one can generalize the very beautiful and simple one-dimensional birth and death process, in which you only have transitions between levels Lk prime R prime, with these states are just up or down one at most at a time. So with that, one can write the infinitesimal generator in this form. I will not go into the details if anybody's interested, but then each of these matrix has also some block structure, and as I've said, this is all guided by the way you've partitioned your states, and of course understanding where the processes are. And then each of these sub-matrices B have also this structure, and as you can see, just depending, the indices are depending on what the transitions are, with the delta, the recycling, and then the internalization and the binding. Yeah. So with this structure of space state, now we can work out a number of things with algorithms that can be solved, not pencil and paper, because of course you have a large number of states, but one can write an exact algorithm, and that's what Martin did. So what are we interested in? So what we're interested in, for example, is 2 stochastic descriptors. The first one is this Ti, which is given a certain state of my process. I am interested in finding what is the first time to reach a certain threshold number of internalized ligands. So if I started with 100, the time to be reduced by a half or a quarter, so that's this random variable. And another, in this case, a continuous random variable. But another descriptor that could be of interest is what is the maximum number of bound complexes B that can be observed simultaneously during the, yes?
In the first model, you have just one type of ligand,
In this case, one type of ligand.
And one of the receptors.
Exactly. Yeah, yeah. If you remember the picture, that picture. Yeah, yeah. I'll talk about the most complex thing so the other thing that one can talk about is the maximum number of bound complexes observed, because that can tell you an idea of how much, for example, competition there is between one ligand or the other, or between one receptor and another. And of course, if you want to study this variable, you can also, or you need to use this auxiliary one, which is the time to reach this threshold number of complexes. And in particular, when this B is the maximum, then you can find the time to the maximum. So these are things that one can do. So just to give a brief description of how one can set up to do this, in the case of the descriptor, which is the first time to reach the threshold number of internalized ligands coming from a certain state, of course, we're only looking at values of N3, internalized ligands that are, of course, smaller than the number I'm looking for, otherwise I'll be wasting my time. And the way one can do this is to introduce the Laplace-Stieltjes transform of my time, and then get all the moments. And I don't think, no, I didn't want to put too many, well, I put some. So the idea is that you can do this, and what you do is you partition your states into the states that, of course, haven't reached the threshold, and the rest of the states just become an absorbing state. So finding this time is nothing else but finding the time to become absorbed, and first step arguments allow you to write these recursive relationships. And what one can do, given the structure of the matrices I've introduced earlier, by this classification of states, these things can be solved, as I've said, sequentially, and if someone, as I've said, is interested, we can show you the algorithms to do so. So the idea is that even though it's computed with a numerical algorithm, it's exact. Furthermore, as I had mentioned, if we keep going, at the end of the day, we run out of ligand if no new ligand is being generated. And one can show, of course, that the late time behavior is absorption into this one-dimensional process in which all that can happen is once you've internalized all the ligands, just have trafficking of the receptors. And important, you can calculate the probability distribution, but also the average number of receptors that you get in the endosome. And this has been useful to then parameterize the model, as I will mention in the next slides. So as someone had asked me, the big picture, or at least the title of my talk was 2 ligands and 2 receptors. So this is the total picture. So the one I've been telling you is this picture A, right? 1 ligand and 1 receptor. And remember, the only one that internalizes is CTLA4. The V is the other receptor, CD28. So the idea is that one can, that's sort of the advantage of doing things with modeling, that you can take each of these little boxes by themselves. So something that we've done is, so box A is what we can do analytically. Then you can add 1 receptor. So you have 2 receptors, A and B, 2 receptors and 1 ligand. Or you can do 1 receptor and 2 ligands, A plus C. Or you can put everything together and you have 2 receptors and 2 ligands. And when we combine all these other modules, what we do is Gillespie simulations. No chance to do this algorithmically. So what have we done about the receptors and the numbers? So we've been very fortunate. There was a group in Oxford with Simon Davis and a modeler in Sweden, Andreas Johnson, and they had provided some of these numbers, and we've also discussed with David Sampson and Lucy Walker at UCL. So let me start with antigen-presenting cells. Antigen-presenting cells double, more or less, the amount of expression, and I'm not talking about the whole cell, I'm only restricting myself to the synapse, the area of the contact between the antigen-presenting cell and the T cell. And then we have numbers of CTLA4 and CD28 in the endosome and in the surface, and the same thing for CD28, but as CD28 doesn't traffic, we have 0 in the endosome always, and the numbers of CD28 don't change between naive T cells and activated T cells. Whereas for CTLA-4, the picture is a bit more complicated, so you've got to start with rather more in the endosome than in the surface for a naive T cell. And as we've not included de novo synthesis of CTLA-4 because we're keeping the numbers of receptors fixed, the trick, at least for today, as I said, is work in progress, is as the cell becomes activated, the numbers of CTLA-4 molecules in the membrane increase almost by, as you can see, by a factor of 10, which is what the immunological evidence seems to support. So at least for today, we will consider these different three scenarios because we don't have the dynamics of synthesis included. These are the binding and unbinding rates of the different reactions for receptors two receptors and two ligands. What we know is that the internalization doesn't seem to be enhanced by being bound to the ligand, and in fact also seems to be independent of the ligand, so that's the assumption we've made. This experimental value that 90% of the CTLA-4 molecules is in the endosome in the absence of ligand allows us to get this relationship between these two rates given by the stationary behavior of the Markov process that I explained, and the fact that it takes about 5 minutes to internalize 80% of the surface of CTLA-4 molecules allows us to get this number for the recycling rate. And of course, as we have also the previous constraint, then we have basically all the numbers. So one can do that, and then let me just walk you through the results. And as I've said, this is all preliminary, but the first thing we wanted to do is if we take CTLA-4 and CD80, this is for a mature T cell talking to, a mature antigen presenting cell talking to a naive T cell that is only expressing 11 CTLA-4 receptors in the synapse. And something that we thought, and what we're plotting here is, this is how many you've got in the surface initially as a function of time, and as you can see, as time's moving, you start losing because of this internalization. So this is a picture of internalization, or the complement of the internalization as a function of time. Something that we thought is, if we could have experimental setups in which we could ask one cell to only express one receptor or one ligand at a time and do all the combinations, as I've said, if we start first with CTLA-4 and CD80, and then we keep adding A is just one receptor, one ligand, we can then go to green, which is add CD28 to the picture, one co-receptor, or add the other ligand, CD86, which is the red, or the whole thing. What you can see is that, of course, when you have everything, things take a bit longer, right? So that's the picture. Important is that the time scales are not crazy. So even though we hadn't done any fitting, because with the experimental data we had, we could get the rates, this is data from the lab, and at least quantitatively, but also in terms of the ratios, this is a cell that is expressing an initial number. So these two plots are experimental data. So as a function of time, what they do is that they're able to fluorescently label the ligand. So the CD86 expressing cell is the sort of the donor cell, the host cell, and CTLA-4 expressing cell is the one that is stealing the ligand. So you can see that very nicely, while the fluorescence is going down in the one, it's going up in the other. And in this picture, what I've done is we have done the simulation for the whole model, so two receptors and two ligands. And in this case, the color coding, I'm plotting is the same thing, how many ligands I start with in my antigen presenting cell, and as a function of time, how much is becoming internalized. And the different colors from purple to yellow is the level of activation of the T cell. And remember that level of activation is enhancing CTLA-4 to move to the membrane. So you start with one cell that only has 11, so that's the purple, versus one that has 110 molecules, so tenfold more. And again, as you can see, the cells that are rather activated internalize much faster. So it seems that at least on a qualitative and quantitative basis were not far off, so the model seems to explain things. What we can also do is, again, look at the distribution of this maximum number of bound complexes, and again, what we've done here is plot the distribution for this maximum. For top one is the naive cell and bottom one is the activated, and of course as the activated is expressing 10 times more CTLA4, if you compare the different models, there's less difference where you have more. Sorry, I forgot to say, and maybe let me just go back, so the other way around. So something that we're currently discussing with the immunologist is the fact that in principle that was something that they couldn't answer. What is more relevant, to change for therapeutic purposes the amount of CD28 or the amount of ligand? So in this case, if I start with just the one simple, one receptor, one ligand, it seems to make a larger effect if I add the other ligand than if I add CD28, for example. So these are things that, as I'm saying, as I speak now, we're trying to put into an immunological context. Why is that the case? And again, if instead of looking at CTLA4 with CD80, I look at the picture with CD86, which of course is expressed at larger numbers, and again, a naive cell versus an activated one, we can again see that the difference between the simple model doesn't really make a difference if I add one receptor or the other ligand, but when the cell is activated, it seems that adding just the one receptor makes less of a difference than adding. Yes, Omar? Previous slide, yes?
One more, sorry. I'm just trying to get some intuition about the time scale there. So it's 50 minutes.
So this is in seconds.
And over there on the right, the date, the experiment.
So this is minutes and this is seconds.
So what I'm trying to understand is, what is the bottleneck rate for that? Because I think you said that the internalization of CTLA4 is very quick, and ligand unbinding is also quite quick, right?
But recycling is very slow.
Okay, so everything accumulates in the sort of recycling state. So the cell has very little cell surface CTLA4, but whenever it gets there, it quickly steals a ligand and goes back in. That's what you're saying. Okay.
Yes. So that's the picture. So what I'll mention now is back to the immunology and where to go from here. I mean, I know that these things are rather painful and I cannot claim I understand them, but something rather important is, can we really dissect these two potential roles of CTLA4? The one that I've been talking about, the stealing of the ligand, and the other one really inhibiting T-cell signal. So the way they can play with it is by having just a control experiment in which you just have normal cells talking to each other, or add anti-CTLA4, and this anti-CTLA4, what it will do is actually block receptors on the membrane, block CTLA4 receptors on the T cell. But you can also add CTLA4 immunoglobulin, and what it does is actually blocking the ligands on the antigen presenting cell. So this experiment, and then you can also block CD28. So what the experiments are showing, that if I'm talking about T cell responses, and for those of you who are not familiar with these plots, you can very nicely label cells with a fluorescent dye CFSC, that when the cell divides, the daughter cell has half of the intensity and the granddaughter cells will have a quarter, and so on and so forth. So the levels of division are encoded by the ratio of this fluorescence, and this peak here is the cells that have not divided, and as you lose intensity, you have these peaks of cells that have divided more times. So you can see that when you block CTLA4, that is the receptor, you still have lots of peaks, so you have an effector response, but when you block with the immunoglobulin, which is really blocking the ligand, you actually stop. So this is supporting the fact that CTLA4 is really not inhibiting the T cell receptor signal, but whereas if you block anti-CD28, you really stop the signal. Then something else that they can do, and again, I'm not going to explain the experiments, but what I thought it would be important to put in the context of what we're doing is that they can look at these systems in time, so they can look at what the levels of expression of the different molecules are at 3 hours and 24 hours. And what they can do is change the ratio in which cells are. So how many, if you know how many donor cells and the ones that can steal the ligand, the recipient, so they can play with the different ratios, and each of these different numbers correspond to different experimental setups. But the idea is that they can play with that, which is something that we can also play numerically. We haven't done that, but that's our plan, to now put different ratios of different cells with these mechanisms and see what happens. So for example, in these experiments, they have 4 cells with ligand to 1 that can steal the ligand, a ratio of 4 to 1 or a ratio of 4 to 4 or 4 to 16. And what you can see, of course, is that that will change the behavior. And then also back to where I wanted to conclude, which is this idea of the role of regulatory T cells, is that they can add to the picture antigen-presenting cells and 2 types of T cells, the ones that are regulatory and the ones that are naive. And remember, the regulatory ones are the ones that express a lot of CTLA-4. So with this, I'll finish and tell you where we want to go. So of course, lots to do. We still need to really fully understand the implications of our results. We want to do a sensitivity analysis of the model, and of course do with a number of restrictions that are, I mean, I would say they're not terribly important, but it would be nice to have a more comprehensive model which can take into account protein synthesis and also the dynamics of the ligand presentation in the antigen presenting cell. And the plan is for our PhD student to make use of the data, and with the knowledge and expertise from John Pop Gosling in Leeds, carry out Bayesian parameter inference and model selection. And back to the picture of this regulatory T cell, let me then explain. So something that it's a lot of ordinary differential equation models have been put forward to the interplay between regulatory T cells and effector cells. So the idea is that these cells called regulatory cells have been called suppressor cells. So the idea is that in an ODE for the evolution of the effector cells, what one has hypothesized is that it depends on how many regulatory T cells, so just like a contact term, not necessarily because these cells come into contact, but because the effect is proportional to how many regulatory cells you've got and to how many effector cells you've got. But the question is, what is this parameter here? So something that we are going to be doing next is, if this is my regulatory T cell, which is the one that expresses a lot of CTLA-4, can I, as a function of the dose of CD80 or CD86 ligand, and as a function of how long these cells are in contact in the synapse due to TCR-mediated interaction, what is the per cell, and as I said, for a given dose of how much CTLA-4 this cell is expressing and how much ligand this cell is expressing, can I get an idea of this parameter, which, as I've said, will depend on the time window of the interaction and the dose or levels of expression. Because as we know, when this APC has seen this regulatory T cell, when it talks to the T cell, it will not deliver signal 2. So this will allow us to properly, from molecular perspective, give us an idea of this time scale. So I'll finish by saying thank you really for the chance to be here today and the Institute for hosting us. Thanks to Luis, who's been leading this work, and Martin for all this new knowledge for us, and experimental support and help and immunological knowledge from Simon Davis in Oxford, Andreas Johnson in Skövde, and Lucy Walker and David Sansom at UCL. Thank you for your patience. And before finishing, just because I realize that maybe some of you will be here later in June, we're rather fortunate that the British Society for Immunology is keen to support mathematical immunology, and there'll be a meeting hosted by Microsoft Research Cambridge, which is just around the corner in the railway station. So that's the webpage. If anybody is interested, registration is open, and if you want to give a talk. And then at Leeds, we host 2 European networks to do with mathematics for health and disease and quantitative immunology. So if anybody's interested to send either a PhD student or a postdoc or yourself to any of the events, we'll be delighted to see you there. Thank you very much.
Let's take a couple of questions. Yeah, please.
So, what's is there some sort of understanding or from your model? What's the role of having two ligands? Like, what if you had just one of them?
That's a good question. So I think it seems to be the case that CD28 is slightly better for one of them, right? So the picture can be this sort of buffering between ligands and receptors. Yet, I think the plan is to do different experiments in which you can really control cells only expressing the one or the other, and then see. I mean, at least the one thing is the difference between CD28 and CTLA4 seems to, I don't know, maybe we have a clearer picture. With the ligands, I don't know so much, but I mean, it could be just a matter of, I mean, which one might be easier to be presented or not, or be more stable.
I just want to understand the experimental evidence to show that CTLA4 is primarily extrinsic from that, I think it was the CFC data. Yeah, this one, yeah.
Yep.
So you said that in I guess, condition 3, you add CTLA4 Ig, so that...
Exactly. So with this, what you do is you block the ligand.
But then don't you block the ligand from binding CD28?
Exactly.
So how do you know that it's not, that it's, CTLA4?
Because of the anti-CTLA4. So when you block the CTLA4 receptor, you don't see a difference.
Right.
So I think that's the evidence. So the hypothesis is CTLA4 inhibiting the signal? And it doesn't seem to be the case, because when you block with anti-CTLA4, you still have whatever your 5 or 6 peaks of division, right?
Okay.
Whereas 3 and 4, as you say, so this is exactly the question. The question is that CD28 still needs to give you signal 2. And only when CTLA4 can do the damage of taking a lot, then it's important. So that's back to, I mean, I don't want to spend too much time with experimental evidence, but then that's when you really go into the other ratios that I was talking about here. And what you can see is that when you've got a ratio that is on the winning side, it doesn't make a difference. But when you can steal a lot of ligand, that is, so it's always this threshold of having too much or too little of either molecule or receptor.
Okay, well, we've got to.
Thank you.
We need